There we go. So this starts with G2 phase. The cell is growing again, prepping for chromosome segregation in M phase, and activating M phase CDK again, which we know was originally the levels were low and everything to get the pre-RCs to form. So M phase CDK and cyclins need to be activated to enter M phase during this process of the cell cycle. And during G2 phase, it's just checking everything, making sure the cell's grown enough, DNA was replicated correctly, no damage is present. So that way then M phase CDK cyclin can be activated and we can continue with the cycle. So we have the beginning of the G2 phase, which is going to be with the G2 checkpoint. So MCDK levels are rising, but they're still currently inactive in G2. So like I said, to enter M phase, we need to get to the point to be able to activate them to go through. So right now we have CAC, and that has phosphorylated MCDK with the activating kinase, so we have CDK and cyclin all together. CAC's down here, and it has that activating phosphate that it's put on. But this one is weaker than the inhibiting phosphate. So at the same time as CAC, WEE1 is the inhibitory kinase that's going to be adding that phosphate onto MCDK. So we've got that one and that one stronger. So right now it's inactive and it needs to be dephosphorylated for this whole thing to become active. So the reasons why we have it inactive right now is, like I said, cell's going to have to grow big enough, make sure there's no damage to be able to continue through everything. So MCDK can't be active quite yet. But when it's all ready, we can go into late G2 phase where we have that positive feedback loop that's going to activate MCDKs to allow it to proceed into M phase. So for this, we have our inactive MCDK right now, and then we've got CDC25, which is going to be the activating phosphatase that's going to remove that inhibitory phosphate. And to get it partially active to begin this whole process is going to be by PLK1, which is just a kinase. So it's going to take off that inhibitory phosphate down here on MCDK that was originally put on by our WEE1. And that's this whole process. And now we have active MCDK. And so once that gets active, you have your positive feedback loop to begin rapid activation of the M-cyclin CDK complexes to build up enough levels of active M-cyclin CDK in the cell to move on to M phase. So that's what we have over here. M-cyclin CDK fully activates CDC25. So like over here, it was just partially activated. Now it's fully activated and it's going to go through and totally remove all of the inhibitory phosphates on inactive MCDK in the cell. So it'll go through, do that. It'll go from inactive to active. And then at the same time of it fully activating CDC25, M-cyclin CDK will also be able to phosphorylate and inactivate WEE1. So then it'll just move pretty quick through because you'll have CDC25 taking all off the inhibitory phosphates and no WEE1 to put them back on. And yeah, that's how you get enough of the M-cyclin CDK active to be able to go into M phase. So there's a couple steps in M phase. This is just a breakdown, little overview of all of the phases that we have. So we've got prophase, and that's going to be that active MCDK that allows it to go into prophase from the G2. You're going to have a breakdown of the nuclear envelope and the chromosomes are going to condense. We'll go into detail with all of these. So then prometaphase will happen after that. You've got the microtubules. They're going to grow and shrink towards the respective locations, and that's going to be going onto the kinetochores, either from like the spindle poles, or some of them are going to overlap. And so they're just doing that during this and making sure everything is properly attached. Then we'll have metaphase over here. So the chromosomes there are going to align on the metaphase plate. The chromosomes will make sure they have all of their attachments and everything, with all of the microtubules going where they need to be again. And then during metaphase, before anaphase, we have the spindle assembly checkpoint. So then anaphase, there's anaphase A and B, but generally it's just going to be the separation of the duplicated chromosomes to their respective poles. And then we have telophase and cytokinesis, where you'll have that nuclear envelope start to reform, as you can see here. And the ring in the middle is going to like separate them off. The chromosomes are going to decondense. The cell is going to divide, and it's going to look back like that again. All right. So next, well, first off, we have prophase. So like I said, we've got that active MCDK, and what that's going to be able to start doing in some more detail is phosphorylate lamins, which are just proteins that are going to be able to break down the nuclear lamina, so that nuclear envelope holding all of the sister chromatids inside is going to be broken down. You can also phosphorylate condensin, another protein that's going to induce chromosome condensation. One way to remember this is the spaghetti to sausage analogy. So these kind of look like spaghetti noodles. And then as they clump up together and like get more towards the center, they'll kind of look more like sausages as the chromosomes will condense. And then it'll also begin to phosphorylate various maps and motors to facilitate meiotic spindle assembly, which is important for prometaphase and metaphase to have all the maps and motors moving correctly and moving stuff around in the cell. So a little overview again of microtubules, and then specifically for the spindle assembly for M phase. We've got asters, which will grow from the spindle poles and attach to the cell cortexes. So that's going to be these green ones. We have the spindle pole here, and they're going to go out towards the cell. And then we have the kinetochore microtubules, which can connect to the kinetochore on the sister chromatids. And those are going to grow from the spindle poles originally. So it's going to be these blue ones that you can see with the spindle pole here going out to the kinetochores that are attached to the sister chromatids. And then you have the interpolar microtubules, which will grow from the spindle poles and just overlap in the middle here, which are these pink ones. So the microtubules will reassemble into spindles and separate the chromosomes during the beginning of M phase by attaching to the kinetochore. And then they're used also in anaphase. They'll be used with separation and movement and everything, and then they'll eventually disassemble during telophase and cytokinesis. So a little bit about the push-pull mechanisms of how these microtubules all kind of work together to move everything around. During these phases, we have three pretty important ones for the cell, and they go along with this diagram. So first off, we have EG5. It's an interpolar microtubule. So if we want to go back, that's going to be the ones that grow from the spindle poles and overlap in the middle here. So they're plus end directed motors, and what they do is they specifically will push the spindle poles apart. So if you want to look, this little gray looking thing is going to be that EG5. Here are the two overlapping microtubules, and then you have some arrows to show that they're going to be pushing away those two spindle poles to their opposite directions. So they specifically walk towards the plus ends on the two overlapping microtubules, kind of like pushing them back as they do that. So the second one is going to be your dynein, which is going to do something similar with the interpolar. It's a minus end directed instead, so it's walking the other way. And while it does that, it's going to pull the spindle fibers together instead. And so it walks towards the minus end of the two interlapping or overlapping microtubules. So for that one, it's going to be this little black guy here. And you see a couple of them and how they instead work in the opposite direction by pulling the spindle fibers together. So these two fibers are getting pulled this way. And then we have another dynein, but it's an aster instead. So that's going to be the ones that go from the spindle poles to the cortexes. So that's going to be a minus end directed motor, and that's going to pull the spindle poles apart. So we can see it's instead the little white character here and how it's pulling this spindle pole away with these arrows and how it walked this direction. Yeah. So after all that, we have metaphase. And just to go over, during S phase, we have those cohesin rings that will form around the replicating DNA during S phase. The rings will keep the DNA strands together throughout and after duplication and into M phase. And that's going to prevent chromosome segregation during spindle assembly and ensure an equal number of chromosomes for division. So everything will stay together during M phase until it needs to be separated. So sister chromatids are secured by those cohesin rings. They're also attached to microtubules at the kinetochore, and equal and opposing pulling and pushing forces will cause them to line up in the middle along the metaphase plate to eventually be separated, which is almost next. But first, we have the spindle assembly checkpoint, which is used to make sure that all the sister chromatids are aligned and the spindles are attached properly to the kinetochore. Make sure just pretty much everything's right, just another checking mechanisms to make sure nothing goes wrong with this whole process. So she briefly mentioned it in class. There's two proteins, MAD and BUB, that are able to sense the microtubule tension on the kinetochores. And then if there's equal tension and they're able to sense all that and everything's good, CDC20 is going to be released. And what basically CDC20 does is it's going to bind and activate APC, which is a ubiquitin ligase. So for this specific example, we have the active APC, and then we have this complex of an inhibitory protein, which you're going to know as securin, and then separase, which is going to be used later on. So CDC20 and APC, that complex is able to target the protein securin and remove it from the situation and go send it off to be degraded. So when that happens and securin is removed from separase, separase becomes active. And the function of separase is pretty much to chop off all of these rings. It'll cleave them and the sister chromatids will be able to separate. So you can see that here, all of these pink rings, separase comes in, and now you're able to have those chromatids separate, go into anaphase, and all of the cleaved cohesin rings in the middle. So that leads you to anaphase. So in anaphase A, we have the chromosomes are pulled towards the poles. The chromosome is like kind of walked by minus undirected motor. So that goes back to like a lot of the push-pull activity you see. But they're basically just moving towards the two different separate poles on the opposite sides of the cell. The kinetochore microtubules are also beginning to shorten with this, which also helps with the pulling process, possibly by this thing called MCAC, which is just an plus end directed destabilizer. So as we know, if it's getting that plus end, it's just going to keep shortening down and down with the destabilizer for the microtubules. And then the spindle poles still stay in their place. They don't totally move yet. It's more everything else that's moving towards them. But then in anaphase B, the spindles will begin to lengthen. The microtubule will grow on the plus end just for this middle pole area. So the poles are going to be pushed and pulled apart, pushed by like the middle areas and then pulled towards the outer ridges of the cell. And then there's going to be that sliding force between the inner polar microtubules from the opposite poles, which is that middle area. So like I said, they're going to be helping push them apart while the other things pull them. And yeah, the pulling force will be directly on the poles to pull them apart, and then everything else will pretty much come with it. So you'll start getting that separation of the two cells, which will lead to telophase and cytokinesis. So we have this great picture here of them almost being separated. So we have the sister chromatids here. They've reached their opposite poles. We now have a cleavage furrow, which is this middle area that's created by a contractile ring. It's just an indentation in the cell's membrane that deepens until basically they split into two nuclei and two cells. And yeah, pretty much it turns into the two cells. And when this all happens, you have to remember M-cyclin CDK levels have now dropped dramatically because we're ending pretty much the cell cycle at this point. So spindle fibers are going to be disassembling now because there's no need for them until they start up this whole process again. The chromosomes are now decondensing from sausages to spaghetti, so just the opposite. And then the nuclear envelope is reforming around the DNA to basically make two identical cells. And now, since we've got low levels of the M-cyclin CDK, we're able to restart the whole process back in G1 with the low levels of CDK because that'll start causing pre-RC formation. And yeah, thanks for coming. I can't see if there were any chats, but do you guys have any questions or anything? Nope, I can